Welcome back to my channel Biochem by World. In today's video, I'll be discussing the structure of eukaryotic mature mRNA. First of all, I'll be giving some of the basic details. Like we know that the RNA is usually transcribed from DNA with the help of RNA polymerase, and the after the we can say the RNA generated after the transcription is the pre-transcript, and it is converted to a mature mRNA through various processes, which is we can term as splicing, or we can say capping, detailing, and a number of other modifications. And first of all, eukaryotic mature mRNA is monocistronic. By monocistronic here I mean that one gene, one polypeptide. We can say the rule is one gene, one polypeptide. That is one particular gene results in the production of one polypeptide. Okay, one gene is going to produce one polypeptide. Okay, and the next thing is the yellow thing here. The yellow marking represents the coding region. The coding region here means that the protein is going to be the RNA, the mRNA is going to be translated from this site, that is the coding region. Okay, fine. And usually this coding region is flanked by untranslated region, UTRs, untranslated region. Towards the here is the 5' end of the mRNA. This is the 3' end. Okay, usually the 5' end UTR is termed as 5' UTR, and 3' end 3' UTR. So the coding region, which is usually flanked with the UTRs, there is a translation start site. The point which the translation starts, it is the point where the ribosome starts adding the amino acids attaching here. Codon is beginning over here. Okay, and basic usually it is AUG. The start codon is AUG, that is AUG is present at the translation start site. Now when the usually the translation takes place in the cytoplasm. Okay, and usually the RNA, or we can say the mRNA, is usually transcribed in the nucleus. Moving from nucleus to cytoplasm, it can encounter a number of enzymes. It can encounter a number of nucleases specifically, I can say, and which can degrade this particular mRNA, and hence the signal from the DNA will be lost. In order to protect the mRNA from this degradation by the nucleases, usually it is capped at the 5' end, and usually a poly A tail is added at the 3' end. Okay, at the 5' end, usually a cap, which is usually 5-methyl guanosine. The cap is 5-methyl guanosine. 5' end is having a cap which is known as M7, that is M7G, 7-methyl guanosine, and at the 3' end usually a poly A tail is added in order to protect this mRNA, in order to protect this signal from the DNA. That was all about the eukaryotic mature mRNA. Thank you so much for watching our channel. Please like, subscribe and share.